All right, we are coming to the conclusion of our very first week together. And now we have micro. The one thing I want you guys to know is to not be intimidated by microbiology. Okay. We're going to relate everything to how it affects us in the operating room. All right. Now, just like with Monday, we're going to briefly kind of go over the Moodle course, and it's only because there's some things that I put in there that I really want y'all to be able to locate. Then after that, we have our chapters. We're jumping right into chapter 1, an intro to micro, and chapter 2, the science behind microbiology. And that's where we're going to explore key concepts and different techniques. Okay. And then chapter 3 is what I really want to kind of hone in on because that is where your um, mini session is in. Okay. I believe we have a video. So let's get started. Now, this is a recap, okay? Your module dates. Our live session is Thursdays, which is today at 5 p.m. Pacific Standard Time. Your office hours are usually right before, and that's only because I'm on East Coast time. So, because I'm in Florida, um, I have it set just an hour prior. But if you need me outside of that, y'all just send me a message for either 200 or 210. I will try to make sure that I can get you in where it's convenient for really both of us with your working schedule as well as getting you in around mine. Okay. With that, there are certain things that I'm just going to reiterate that you already know. Make sure that you knock out those 2 attempts on your knowledge checks. It's really going to help, especially with micro. All right. Because this is not one of those subjects where you're just like, huh, I know it all. And I get that. All of your assignments, make sure they're in by Sunday, 11:59 of that week because they are going to close. And if something happens before Sunday, let's say today is Thursday and your computer goes down, let me know as soon as possible. It's a lot easier to kind of work with you before the due date because after everything closes, it's almost going to be impossible to get anything open unless something crazy happens, right? Like a case by case basis. All right. Now, for our class. Again, just my information, office hours, whenever we do set up a time, even if it's outside of normal office hours, you're still going to go ahead and click this link and then we'll link up. You can email me. Again, text is usually the best because I usually have my phone near me. And one of the things that you'll notice, especially in week 1, is we have your regular introductions, okay? Where I kind of tell you what's gonna really gonna happen in the live sessions. I try to make them a little bit different from what you're used to seeing. And then your mini sessions. These are going to be really important because I am going to be sprinkling a lot of that information into our live sessions. And you're going to see that this evening. Now, even if you haven't had the opportunity to watch the mini session, don't worry. I'm going to be covering different things, but moving forward, it'll be a lot easier because then you'll feel more comfortable as we're going through that material. Okay. As far as our syllabus, it looks pretty much just like with 200, all right, where we talk about the books and the information that you're actually going to get out of this course. Okay. And I really just wanted to make sure you guys knew exactly how to get to those mini sessions because I do reference them a lot throughout this module. Now, before we even get started, I want to turn it over to you. What are you expecting from this particular course? Not the module, but from microbiology. And then, if you would like to kind of give me some ideas of what you would like to see. And the reason I do that is because I do like to cater my classes to the cohorts. Okay. So you guys can be typing that in. What is something that you're expecting to learn from this course? Everything, honestly, I don't remember anything about micro. I love that. I love honesty. That's perfect. That is perfect. And we're going to be breaking down a lot of different things. I'm not sure, but I do want to learn something new every day. I like that. Microorganisms and how they work and where they're located. We're definitely going to go through all of that. And you'll be surprised maybe on how much this relates to what we do in the operating room. When I say we're the patient's advocates, I mean on every single level. I'm excited, whatever comes, I'm ready. I love that. Now, and you guys can keep typing it in. I'm going to be reading them um, as we go through. So we have our introduction to microbiology. Now, this is where we become interactive. Whatever you're doing, except if you're driving or if you're at work, I totally get it. You can do this later. I want you to grab a sheet of paper, open up notes if you're on your laptop, your iPad, and label 1 through 10. Okay. And if you've watched the mini session, you kind of have an idea about what we're going to do. As we go through the live session, I want you to write down the 10 reasons why it is important for you to understand microbiology. So I asked you guys in the beginning, and then as we go through this introductory live session, you may be surprised as to what those reasons are. And at the beginning of next week, everyone is going to type in their number one reason why it's important for us as future surgical technologists to study microbiology. Okay. With that, every live session, every chapter, we have learning objectives. It is the nature of the beast. So we have key terms, which we're going to have every live session, but we are going to discuss the responsibilities of what we do and other sterile members in the prevention of disease transmission. We're going to start off talking about significant historical contributions from pioneers and bring it all together to the 20th century, to our current public health concerns. But we're not done. We also have to explain, as you see on the slide, spontaneous generation and abiogenesis, where they were disproved, and the impact on the work of future researchers. We're going to look at some pioneers, and then we're going to bring it all together with some critical thinking. And that's just in the first chapter. With microbiology, it's really cool because they do what's called a clinical significant topic. Now, we rely heavily on scientific research and the cumulative knowledge of the scientists throughout the centuries who have either created the foundation for our ability to recognize sources of infection, and think about what we do, developed effective preventative measures to minimize the harm to our patients and ourselves from the multiple of microbial pathogens. And sterility cannot be seen, so adherence to the principles and practices of asepsis and sterile technique are so important in just reducing the potential for surgical site infections. But you're going to start hearing me say SSIs and HAIs. So SSIs, surgical site infections, and HAIs, hospital-acquired infections. Also, transmission cannot always be prevented, unfortunately, but all available measures should be utilized for breaking the chain of infection. And that is this week's clinical significant topic. Now, as STs, our main job is to make sure that our patients get the best care during surgery. So you guys are going to be working in hospital operating rooms, hybrid rooms, radiology suites, outpatient, or even doctor-owned surgery centers. So this is where the pavement meets the road. Surgical conscience is the guiding rule of our work. It's not something that we can just train you on. It's something that you have to kind of adapt and make your own. It means always reporting any mistakes in maintaining sterility, even if no one noticed, and sticking to the best practices to keep the patient safe. Now, learning microbiology, hint, hint, helps you understand how to prevent infections and protect patients during surgery. So keep in mind, you're still writing those 10 reasons why it's important for you to understand microbiology. Now, talking about surgical conscience, we have to be able to recognize correct breaks in sterile technique, whether they are committed in the presence of others or alone. So let me break this down. We have to be able to spot and fix any mistake in sterile technique, whether they happen with the entire room there or if we are there by ourselves. So here's a quick video on surgical conscience that I think ties right along with what we're talking about. Let's take a look.
If you're a surgical student, mastering surgical conscience is a must. Here's why. Surgical conscience is all about maintaining the highest standards of cleanliness, honesty, and responsibility in the operating room. It means speaking up if you see something wrong, even if it's uncomfortable. Think of it as your inner compass guiding you to do what's right, no matter the pressure. Why does it matter? Because patient safety depends on it. A single lapse can lead to infections, complications, or worse. So, always double-check sterilization, follow protocols, and never cut corners. Remember, surgical conscience starts with you. Practice it daily, ask questions, and learn from your mentors. It's not just a skill, it's a mindset that will shape your entire career. Stay sharp, stay responsible, and always be the advocate your patients need. This is where surgical conscience starts. Remember, you are the patient's advocate.
All right. What did you get out of that quick little video? How did it resonate with you as future surgical technologists? Accountability. We have a lot of responsibilities to protect the patient. We are the patient's advocate, even when it can be uncomfortable. And at times, it can definitely be uncomfortable. True story. I was working in ENT at a children's hospital in San Diego. And one of the surgeons, we were doing an LTR, or we were doing a case, because I got to remember where you guys are. We were doing a case where we were dirty and clean, meaning we were doing part of the case in the mouth, which is just surgically clean. And we were also doing a part where we were actually going into the larynx, which was sterile. So one of our biggest roles is making sure that things don't cross-contaminate. So when the surgeon was going into the mouth, I had to make sure that particular surgeon had extra gloves, because when they would come back around to the sterile field, I am not going to let you be all up in the patient's sterile throat after you just came out of their clean mouth. And so some of the times I had to say, hey, doc, let me change your gloves. Did that particular doc get a little bit frustrated? Of course, because I'm slowing the case down. But at the same time, we are that patient's advocate. Make sense? All right. Now, the other thing we talk about is the transmission of disease. It cannot always be prevented. Okay. That was one of our learning objectives. But those of us that have worked in healthcare, we have tools at our disposal that allow us to simultaneously protect ourselves from exposure to patients, and patients from exposure to personnel. These tools we're going to use. And in the operating room, it is second nature. We're using gowns and gloves and masks, shoes, goggles. For those of us that wear glasses, we do have shields for if you don't want to take off your glasses, but we also have masks that have the shield connected to the mask. I'm letting you know you're going to want to have some type of eye protection on. Of course, as a student, you're going to be required, but when you're in the field, of course, you're doing your thing. But keep in mind that more things can splash up and around than you think. Picture it. You're 5 years into your career, okay? And you're doing a shoulder arthroscopy, which is just a shoulder case, and we're taking camera inside the shoulder. Now, in order for that case to be successful, we connect a camera, we have fluid that is going in to help us to be able to see the joint better. So in the abdomen, I want you to think we use gas to blow it up, but in the shoulder and the joints, we use fluid, okay? Now, during this case, you are asked to give the drill to the resident. You take a deep breath and you give it to them, knowing that you're going to be watching them. And as they're drilling, whatever was in that bone is coming out of the trocar. And the trocar is what we use to kind of place our instruments in and out. And a lot of times we have multiple trocars, especially in shoulder cases. So just think about it. You forget to wear your protection for whatever reason. Where do you think all the juices and body fluids and whatever else that is splashing out of that shoulder could end up? Right? So this is why we, like, I'm the type like, okay, but why are we doing it? Right? Well, this is why, because I have been in cases where a student or a resident did not have on protection and they had to go and flush their eyes out. And I'm just looking like, because it can happen. So we're going to protect ourselves. The other thing is breaking the chain of disease transmission. So we have standard precautions. These practices apply to all patients, no matter their infectious status, to reduce the risk of infection spread. But what does this mean for us? In the operating room, we treat every patient as if they have everything. Now, that's not saying that we're like, oh my gosh, stay away. No, no, no. What I'm saying is we are going to be protected no matter what. We are going to always wear shoe covers or assigned shoes for the operating room. So you don't necessarily have to wear shoe covers, just so you guys know. You can have shoes designated for your operating room once you get into your field that do not come home. Because one, you don't want to bring that stuff home, okay? We're going to always wear gloves. I double glove. That's just me. It gives me just an extra sense of protection. You're going to always wear your gown, hair cover, mask, face cover, shield. Okay. That's just things we normally do. Hand washing is really big. Of course, you've seen the fancy hand washing that we do in the operating room. And module 4, we're going to show you how to do that. But besides that, we wash our hands with soap and water. We use what's called an alcohol-based hand sanitizer, kind of like the ones you see where you can get from anywhere. But we use a specific kind and we still kind of scrub it in a way that it kind of protects us. Okay. And then I already mentioned our PPE. We look at transmission-based precautions. We do that as well. These are just going to be extra precautions based on how the infection is spread. This helps stop the infection from moving from people to person or person to person. Now, sometimes we have patients come in and they have precautions that we already know. So we still have our gloves, we still have our gowns because we're still going to avoid spreading the infection. We also get certain masks. There are times when you'll be assigned a case and there's droplet precautions. So we have certain masks, we all get fitted so that if we need them, we grab them. And we have signs put on the outside of the door so anyone coming in or out of our operating room suite, which we try to minimize it, they know as well. Now, wearing masks does prevent spread of infection. The one thing that I'm going to tell you that it's going to be really awkward for you. Let's say you do have to sneeze, and I'm going to break it down so it makes sense. I'm going to ask you guys a question, what you think. So when we talk about the sterile field, that is the surgical incision where we're cutting. Okay, so that's our sterile field. You have on your mask. If you're going to sneeze, what direction do you think you would normally sneeze at?
Away
Are
from
you
the
going,
patient.
ah, actually, and I love that you said that. you're going to sneeze towards the patient. Why do you think that is? Right? I know. I see your face. Do I have a mask here? I don't. Okay. It's over there. You have on your mask. If you turn away from the patient, you still have a little gap. So, what happens to that field? So, a lot of times, what I do is if I have to sneeze, try not to. If I have to sneeze, I step back, sneeze, hopefully nothing else coming out, and then we keep moving. Does that make sense though? Now, book versus real life. Real life, you're going to see some stuff, right? But just know proper way is if you can remember when you are sneezing or if you have to cough, you're coughing towards the field. You usually step back and then you cough towards the field. It's a little bit unorthodox or something that you're not going to be used to. You're also not doing any of this, which is going to also be different because you don't want to contaminate. Okay. This isn't going to be sterile, but your hands and gloves are. So, you know how we're normally like turning, you're not doing any of that. You're standing straight, stepping back, and coughing. Okay. So, I like that step back technique. Noted. I love it. All right. Some of the stuff we have. So these are these masks I was telling you about that you do get fitted, especially at certain hospitals. At least at the Children's Hospital, we all got fitted. So, like, let's say we did have a patient that had any type of droplet, we would be assigned and we can go grab our size and we have them in the operating room. But you have cover gowns that we use before we open our case. We take our cover gowns off when we're getting ready to scrub our hands, but you have your gown, your glove. I don't know if you can kind of see, but this has the combination of the mask and the shield that I was telling you about. But you still have the option of wearing face shields. We have beard um, mask as well for guys that have beards. So they would wear the regular mask and then the beard mask as well, right? Because we don't want anything dropping into, the field, which is the sterile field is what again? Do you guys remember? What is our sterile field?
Where you're cutting.
That is right. That is where we're cutting. I love it. Okay. Now, the science of microbiology has had its most significant early impact on human health, uncovering the cause of major killers of the day and the methods to treat them. I want you to picture this. Back in the day, when barbers were surgeons because they knew how to cut hair, they also didn't really believe in washing their hands because they felt that washing their hands caused germs and infection. They would also go from a patient that had just died from some crazy disease to treating other patients, and then that patient would get sick. Now, the midwives were the ones that had the most healthy patients. Not saying that their patients didn't succumb to certain diseases as well, but the rate was really low. And the reason was because they believed in washing their hands in between patients. Okay. Now, several scientists have actually shaped our understanding of infections and immunity. I'm going to try to say their names without messing up. Girolamo Fracastro. In 1530, he was the first to publish research about syphilis, theorizing that organisms should spread or could spread through direct or indirect contact and even in the air. And that was back in 1530. Robert Hooke in the 1600s built the first compound microscope that we're going to take a look at in this live session, really small, and was the first to use the term cell in biology. Antoine van Leeuwenhoek in 1673 observed that and drew bacteria and protozoa for the first time, providing an accurate illustration of these microorganisms. Francesca Redi conducted experiments with meat to disprove the idea that life spontaneously appears, providing strong evidence that living organisms come from other living organisms. Edward Jenner the first to use vaccination to prove immunity. He successfully inoculated an 8-year-old boy with cowpox, and it actually protected him from smallpox. And then in the 1840s, I needed to kind of separate this. In the 1840s, Ignaz Semmelweis observed a high maternal death rate from what was called puerperal fever and realized that there was an importance of none other than hand washing. Now, keep in mind how many years that they were not hand washing. 1840, even though the midwives, patients, were more successful. All right. But here's the problem. Unfortunately, other doctors didn't follow his advice, and Semmelweis died from a similar infection in 1885. So, unfortunately, his doctor that he had did that same practice that I mentioned. He got sick and died, even though he was showing, hey, look, here is evidence showing that washing hands prevents. Isn't that kind of crazy? Like that is crazy to me. And then John Needham and Lazario Spallanzani, these were both priests. They studied spontaneous generation. Needham believed in a vital force that created life, while Spallanzani refused this with experiments showing that life doesn't appear without other life. Okay, so these are just a couple of pioneers. One that I'm not sure if you have heard of is Louis Pasteur. In the mid 1800s, his experiments disproved again, talking about the vital force, and he actually came up with the germ theory of fermentation and later the germ theory of disease. He is actually best known, and you can kind of see here from the Getty image, um, for his discovery of the link between yeasts and alcohol, which gave scientists a new point of view of bacteria and the difference between aerobic without air and anaerobic, or aerobic with air, anaerobic without air types. There was a proof of relationship between food spoilage and microorganisms, which could be prevented by just low heating to destroy the bacteria without changing the flavor of the food. So basically, cooking the food all the way through. Now, alcohol or milk products kind of came through with a process termed as pasteurization. Discovery of the protozoan as the etiology, or we call it the cause, of a silkworm disease. He also had the use of rabies vaccine on children, taking a risk to his professional license. So he was just like, I'm going to risk it all and show y'all this works. We have Joseph Lister. He was a surgeon who, in the late 1860s, embraced Pasteur's link between microbes and disease in animals. And then he took it a next step. He also established the first principle of what we do in the operating room day in and day out with medical practices known as aseptic technique to really protect us, our doctors, from pathogens, microbes, by the use of phenol, which is carbolic acid. And they use that as a skin scrub, kind of tough on the skin, but it kind of made a way for what we use now. Now, they had it for pre op, skin prep, they had it on dressings, and they even had it in an aerosolized spray that they would put in the operating room. He is also known, and you may see this question somewhere. I've seen it on CST exams, I've seen it on week one. He is known as the father of modern medicine, or father of modern surgery, I should say. And that was because of his insistence on surgical cleanliness, the use of what we do now: surgical gowns, gloves, disinfection, boiling of instruments between procedures. At that time, that's what they had. They didn't have autoclaves and everything like that, but boiling the instruments in between procedures. We get into the golden age of microbiology, continuing on with great I would say um, scientists, okay? Robert Koch. In 1876, he became famous for discovering that rod-shaped bacilli bacteria were the cause of anthrax in cattle. He also developed what's called Koch's postulates, which is a step of experimental steps that he did to link specific microorganisms to specific disease that we still use today in microbiology, like cultures. There are many times in the operating room where our circulator will give us a culture that will hand to the surgeon. They will do what they need to do with that culture, hand it back to the circulator, and send it off to pathology. Now, we talked about a lot of individuals, but we're not done. First individual I want to bring up is Ruth Ella Moore. In 1933, she was the first African American to receive a PhD in bacteriology. And in 1936, she joined the American Society of Microbiology that led to extensive body of research and eventual treatment of tuberculosis, the second highest cause of death in the United States at that time. Publications on dental caries and blood type associated diseases was also one of her big things that she came up with. Elizabeth Bougie. In 1944, she was part of a team of Rutgers University research scientists who developed an antibiotic, streptomycin, used to treat, again, deadly tuberculosis and other serious infections. Other male team members were awarded the Nobel Prize for Medicine, but the lead team scientists, they kind of left her name off the patent because she would only get married and have a family. So they felt that she didn't really need that recognition. We also have June Almeida. She is a Scottish born immigrant to Canada who never completed her undergraduate degree, but became a skilled lab tech. She gained professional recognition for her techniques for identification of microscopic particles, the development of the use of antibodies to viruses in the lab under magnification, and at a London medical school after she returned, went back to London. The first time identification of the coronavirus in 1964, the same type of viral pathogen responsible for none other than COVID-19, was discovered through her skill in the use of electron microscopes. So these are a lot of individuals that have kind of fast forwarded and made it possible for us to be able to protect our patients and be true advocates for not only our patients, but ourselves. With that, over the past 120 years, discoveries in microbiology, bacteriology, virology, and genetics seem to have developed exponentially and have continued into the 21st century. Now, a study of the timeline of those years is what you see in chapter 1, and it teaches us about the microbial worlds and how incredibly resourceful all of these microbes and bacteria are in survival method. And for the good or bad, the human race, we have to find a way to coexist with these viruses and bacteria and so on. So we have to recognize the enormous strides that have been made in science in, believe it or not, a relatively short period of time, and how closely we are interconnected with each other, with humanity and our planet. All right, let's bring it back to us. The last part of the chapter talks about standard precautions. We have tools, like I mentioned, to protect us. Okay. Knowing how disease spread is really helpful, but it's even more important for us to follow these guidelines. Now, the CDC has special committees that create standard precautions or have created standard precautions that we use day in and day out. So, we're talking about treating all blood, bodily fluid, and other materials except sweat as if they are infectious. And that's what we do in the operating room. That is why I choose to always double glove. Okay. Now, these precautions apply, like I mentioned, to all patients, no matter if they are known to have an infection or not. So then in our mindset, it doesn't matter because we're going to be as stringent with that patient that we know has a contact precaution as with that patient that we don't know has a contact precaution. So this is why we are very stringent on our hand hygiene, why we always wear our gloves and our gowns and our mask and our eye protection and face shield or safety goggles. If you have your own safety goggles, once you get in the field, you can wear them. We work on safe injection practices. We look at proper handling and cleaning equipment. I'm going to show you in this course diseases that patients have gotten that in some cases are fatal from just improper technique. And we're going to get really into it when we talk about ophthalmics and the diseases that can occur where a patient becomes blind from, bacteria and pathogens that are unfortunately not, how do I say, taken care of before that instrument has made contact. Okay. Now, when a patient's specific infection is known, there are additional transmission based precautions, but we're still going to treat every patient as if they have everything. We're going to be looking at respiratory hygiene. That's why I mentioned cough etiquette. Slightly different when you're scrubbed in as when you're not gowned and gloved and everything. We're going to talk about how to actually get needles from the field to the surgeon and back. We know that masks in the operating room are extremely important because we're right over that surgical field where the patient is open. We see their insides. Okay. What questions do you have in the very first chapter? Does that make sense before we get into chapter 2? Yep. Okay. Shifting gears a little bit. Now we're going to get into micro. All right. And I get it. Sometimes this material is not the most exciting, but we're going to do our best. Okay. We're going to talk about the tiny, invisible world where the real superheroes live. It is where bacterial viruses and other microscopic creatures, they come in and they just run the show. They're sneaky, they're powerful, and believe it or not, unfortunately, they are essential to life as we know it. So again, we have some learning objectives, pretty much the same. Looking at terms, normal flora is really big because we all have normal flora on us. We're going to talk about that. We're going to look at classifications. We're going to talk about prokaryotes. We're going to talk about prions. We have to look at nomenclature, taxonomy, and then again, bring it all together so it makes sense for why it's important to us. I hope you guys are still writing this down. So, what is microbiology? Well, as the slide says, it's the study of tiny life in forms like bacteria, fungi, and viruses. It focuses on how they work, how we classify them, and how we can use or control them. In the operating room, understanding microbiology does help us to prevent SSIs. Remember, I was talking about that little microscope? Well, there it is on the screen. Now, microbes are also known as microorganisms, which include microscopic living organisms, bites of material that are not alive. Here is that famous Van Leeuwenhoek's microscope that provided a means to observe the otherwise unseen microbes. Now, think about the microscopes that we have today. Do you see how tiny that microscope is? Imagine using that day in and day out as a pathologist. Now, modern age advances in microscopy, I know, right, have allowed researchers to study the tiniest of pathogens, including viruses, which are with, I should say, electron microscopes and specialized techniques. The explosion of research requires a method of just classifying the organisms and establishing standardized names for consistency across scientific fields. Again, microbes are found on every area of the planet and comprise the largest group of inhabitants going back approximately 3 billion years. No living beings can survive without some exposure to or relationship with microorganisms known as its normal flora or its microbiome. Now, when even normal flora organisms escape their typical environment, unfortunately, they are capable of causing serious pathological conditions. Non-living viruses and prions require mechanisms of what we call replication other than just normal reproduction. And we're going to talk about that. Viruses, they go in and they penetrate healthy cells to become parasites and part of the cells spreading during reproductive cellular process. So they're kind of like hidden and then they go in and manipulate it. And then now that manipulation starts to double. We get into Aristotle's system. Okay. Now, this is going to be important. You guys are going to have to know these. Scientific classifications have been used since ancient times. In the 4th century BCE, Aristotle classified living things into two kingdoms, plants or plantae, and animals, animalia. This system was used for centuries. But in 1894, Ernest Haeckel added a third classification called the kingdom protista, which included both single celled eukaryotes and bacterial prokaryotes. Okay. In 1956, Herbert Copeland split kingdom protista into two, making a fourth kingdom. So we're just adding on and splitting and adding on and splitting so that there was more clear separation of single celled eukaryotes from bacteria. But we're not done splitting yet, y'all. In 1959, Robert Whittaker proposed 5 kingdom classification system, later revised in 1969. His classification included Monaria, protist, fungi, plants, and animals. You guys thought we were done, huh? Nope. In 1977, Carol Woos and his team discovered a new group of microbes called archaeas. These microbes are different from both eukaryotes, like plants and animals, and prokaryotes, like bacteria, but they share a common ancestor. Now, archaea are often found in extreme environments like glaciers or hot springs. They resemble eukaryotes more closely and may be the ancient ancestors of actual both groups. In 1990, Dr. Woos proposed a new classification system known as the three domain system, which included bacteria, like simple celled organisms found everywhere, even on our bodies. Some of the characteristics, they're going to lack a nucleus. Some do cause infections, while others help with digestion. Archaea, which are single celled organisms that live in extreme environments like you see in front of you, hot springs. They have different chemical makeup, and it helped scientists really understand how life can survive in extreme conditions. And then eukaryotes. These are organisms that have complex cells that have a nucleus, plant, animals, and fungi. Now, their cells have membrane bond organelles and can be single celled or multicellular. We're not done. In 2010, scientists were like, we're going to question these three domains, not really feeling them. And they found that the genetic sequencing of these groups didn't include non-living microbes like viruses. So again, in 2010, the researchers proposed a new system called the two empire system, where you have separate living domains. You have the three domains, and then from non-living viruses. This non-living group includes the plasmids, see if they're plasmids, prions, transposons, and viroids. They discovered that there are far more viruses than there are microbes in the living cellular group. So then you have your eukaryotes, cell membranes bound organelles, and a nucleus with the DNA. Prokaryotes, cells contain genetic material in protoplasms, but not in the nucleus. Of course, again, your archaea. So you kind of see that down here. And now we're done with the systems. All right. We now look at viruses. What are they? They are tiny agents made of either DNA or RNA, which is just your genetic makeup. So DNA and RNA are your genetic makeup. They are so small that you can only see them with an electron microscope. Now, the genetic material is protected by like a shell called a capsid, which is made of proteins. These proteins are known as capsomeres. Okay. Now, viruses are considered non living because they can't carry out life process on their own, like reproduction or metabolism. They are acellular, meaning they do not have cells. So you guys should be asking yourselves, well, then how do viruses reproduce? Haha. Viruses cannot reproduce by themselves. Instead, they're going in like a sneaky person and they invade living cells and hijack the cells' machinery to make copies of themselves. Because of this, they are called obligate intercellular parasites. They have to have a host to replicate and spread. The chapter also wants you to know about viroids. They are even smaller than viruses and are believed to be a simpler form that might have come before what we know as the modern viruses. They consist only of a small loop RNA, which is, remember, that part of the genetic DNA, and do not have a protective protein coat. Now, when a viroid enters a cell, they go in and they interfere with the cell's own genetic makeup, RNA or DNA. And then what that does is disrupts its function. This can lead to diseases in plants and possibly in other organisms. We look at prions, and this is big, especially when we talk about brain injuries and brain diseases. Okay. And so that's why you kind of see the image of a brain up there. Prions are non-living agents that cause serious and often fatal diseases in the brain. They are linked to diseases known as transmittable spongiform encephalitis, TSEs, which affect the central nervous system. Now, disease caused by prions in humans like CJD, which we're going to talk about, which is Creutzfeldt-Jakob disease, and its variants. In cattle, it's considered mad cow disease, and it's bovine spongiform encephalitis. In sheep, it's called scrappy. In deer and elk, it's chronic wasting disease, or CWD, in North America. Now, how do these prions work and how do they cause this issue? Normal proteins, and that's why I have this right here. You have your normal proteins, okay? When the prion will come in, what it does is it will fold have an incorrect shape called a PRPSC, going too deep, but just know that it folds into abnormal shapes. These abnormal proteins then cause other normal proteins to also fold. So it's kind of like show one and then they start doing the same. And if you notice, these prions have gone into the normal proteins, kind of linked with them, folded, and now they think they need to fold too. Does that make sense? A little bit? Okay. So when we're talking about the brain, okay, the misfolding leads to the destruction of brain tissue. So think of these normal protein now in the brain. Okay. And now these prions are coming in and destroying the regular protein, and it's creating holes that give the brain a sponge like appearance. There are other diseases that we're going to talk about in this particular module. Some are rare inherited conditions causing issues with like coordination and memory. There's one that's fatal familial insomnia. It's a rare genetic disorder where prions, okay, prions, think of the squares, disrupt sleep patterns. There's another one, Kuru, which the chapter talks about, is a disease caused by eating infected human brain tissue. So if you've ever heard of like any individuals that like have brain as a delicacy, just keep that in mind. This was a ritual in New Guinea. Biggest thing to know is prions are just misfolded proteins. So think of a normal protein as something that's round, it's doing what it's supposed to do. And then these misfolded proteins cause severe brain diseases by creating more misfolded proteins. And that's what really leads to the brain damage and the significant health problems. Does that make a little bit more sense? Okay. We also talk about taxonomy. Okay. The system of just classifying, naming our organisms. All right. Species, genus, family, order, just like you see on the slide, as well as our nomenclature. Okay. Now, with taxonomy, we're looking at kingdom, phylum, kind of like we learned when we were younger: kingdom, phylum, class, order, family, genus. And then species, which is the most specific group. For prokaryotes, the classification system used in what's called the Bergey's Manual of Systematic Bacteriology was a standard reference just for microbiologists, just kind of food to know, okay? And it is available for anybody who wants to dive in deeper into microbiology. We also get viruses, and they're going to be classified on a couple several factors. We look at their shape, so we look at morphology, how it looks. We look at the nucleic acid type. Does it have either DNA or RNA? Remember, that's just genetic material. We look at the mode of how do they replicate. Okay. We talk about the types of hosts and organisms, and does it affect animals? Does it affect plants? Does it affect bacteria? And then we look at types of diseases caused. So these are just how we look and categorize viruses. Believe it or not, we are pretty much at the end. Okay, so if you had the opportunity to watch that mini session, you know what this is about. This is coming from chapter 3. All right. Now, you've got a couple of media up on the screen, and you've got a couple answers on the bottom. The question I have for you is, do you think you can correctly identify them? Does not look like we're going to get to our Quizlet, y'all, because I'm not going to keep us later. So the Quizlet is going to be in the archive, and it's going to have terms from all 3 chapters on it. And it is another resource to use as we get ready for that midterm. Okay. But if you have figured it out, we're just going to kind of go through it because we are kind of short on time. I will try to timestamp these. So if you want to give it, a try later, you will have the opportunity. So you have A, B, C, D, E, and F. You have the terms at the bottom. Okay. Again, I'm going to time stamp it. And then this is the correct answers. Okay. Like I mentioned, we are, and you can take a screenshot if you guys want. Like again, like I said, I'm going to try to timestamp, not I'm going to try, I'm going to timestamp this particular live archive. I like that. I got 3 right. I love it. All right. Again, here is the link to the Quizlet. Don't stress. I'm going to also put it in that archive like I mentioned to everybody. Okay. All right. And then keep in mind, we're going to share this next week. Your number one reason as to why we care. Okay. So this will be one of the first screens that we see on week 2's live session. Why do we care? Why is this important for us? We covered a lot of different topics this evening. Have another challenge for y'all. If you guys remember from the micro or the mini session, we also had a little microscope we had to look at. Again, because of time constraints, it's going to be kind of quick, but you guys can always pause. So you'll have a timestamp for the media, you'll have a timestamp for the microscope as well. Because these are the correct answers. Keep in mind that understanding the parts of the microscope is going to be important. There are certain cases where we do have the microscope. A lot of colonoscopies and EGD, they may have the microscope in, and we may be the one kind of setting it up for our surgeons. So just kind of keep that in mind. So it's really important just to understand different areas. Sometimes you may need to focus for them, clean it, different stuff like that. Okay. What did we learn? First week, I feel like is always a little bit longer. You're going to notice that we are going to be a little bit faster. But for this week's discussion forum, for those of you who are not here, I want you to identify the items on the microscopes number 3, number 9, and number 7. Post your answers in the forum. Also, I want you to consider how can applying microbiology principles in our surgical setting help reduce the risk of HAIs? What role does microbiology play in supporting these efforts? Let's discuss that in the discussion forum. You guys, we talked about a lot this evening. I talked a lot, and don't worry, as we get forward, we're going to have a lot more conversation because we're going to be seeing a lot more things, and you guys are going to be kind of looking at stuff. And my whole goal is to think outside the box. How does it affect us, and how will it affect our patients? Okay. So we talked about terms in both chapters. We looked at responsibilities. We talked about the different microbes. We talked about the entrepreneurs or the ones who came in and discovered different things to help us be where we are today. Chapter 2, there was also more terms, but we got a little bit more with normal flora, which is actually on our bodies, which we're going to get into a little bit more in this course. We looked at prokaryotes and eukaryotes. We're going to talk a lot more about those, as well as taxonomy. And we brought it back to why it's important to us, which we're going to talk about first thing next week. In the mini session, it's all about the lab, from different culture media, from gram staining, from gram tests, and a lot of different microorganisms. With that, what questions do you have for me? No questions right now? That's okay. If something does arise, y'all, please let me know, because I really want y'all to understand micro, not be intimidated by micro, and just knock it out of the park. With that, I do want to wish you guys all a wonderful rest of your evening. Have a wonderful and safe weekend. And I look forward to seeing everyone who can attend next week's live session. Good night, everybody.
No, thank you.
Thank you. Good night.